Hi, everyone. My name is Max Foreback. I am here with Michigan State University, and I will be presenting my work with my co-authors, Leither and Emily Dolson, on the role of abiotic parameters for the promotion of egalitarian major evolutionary transitions. It's a pretty loaded title, and I'll probably spend most of this talk really digging into the system we used, what these words mean in the context of the system, and then at the end, we'll round out with some brief results and future directions. But to start out, we'll just do a brief journey into definition land, where we can kind of talk a little bit more about what a major transition means and how we thought about it in our system. And broadly, major transitions are concerned with changing the level of an individual. I'm a big examples guy, so I'm going to run through a quick example that kind of shows a major transition in action in a very, very, very simple system with just three things interacting. And these three things are maybe single-celled organisms. We have the heart, the smiley face, and the star, of course. And they're growing, they're moving around, they're competing, whatever. It doesn't really matter. But over time, they're changing their concentrations, they're changing where they are in the world, and they're growing. It's not the right slide. Okay. Anyways. If I give you more information about this system and I say that the heart and the smiley face actually grow really well together. When they're in the same cell, they can grow very fast. And I'll go even further and I'll say that one of these things will excrete a membrane that will encapsulate the other when they're at a high enough level. With that information, we can imagine a system where a scenario in which this heart and this smiley face within the membrane grow so much faster that they can outcompete the neighboring cells, and they move about the world, and they eventually will dominate. And so now when you look at the system, if you look at this end state here, it looks far less like we have three individual constituent parts and far more likely like we have one low-level unit, the star, and one higher-level unit composed of those initial units, the heart and the smiley face. And so here we've seen kind of a situation where you started out with three lower level units, and over time, due to how well they work together, and this extra membrane that we've kind of added as a simplifier, that we now have two units at the end. So we've changed what it means to be an individual here. The heart and smiley face no longer are individuals, they are one individual within the membrane. And so how do we actually study major transitions? Well, my lab does a lot of ecology and evolution work. And so we constructed an artificial ecology simulation composed of a grid of cells. I'm going to call them cells. They're not related to biological cells in any amount, but they are just regions of local space. And I'll also be calling those shapes we were talking about earlier types. I talked about them as single-celled organisms earlier, but really what's important to us now is that they're just things that grow and or die based on interactions with other things. So they could be biological species. They could be, we talk about them in our paper as autocatalytic chemical reaction networks, if there are any origin of life researchers out there. But the important part is that they just grow and die based on interactions with each other. And so we throw them in our world, and this is our artificial ecology. It's a grid of cells containing these things that interact with each other. This is what it looks like in actuality without the fun cartoon shapes. We'll go back to the cartoon shapes in a bit, but this is an actual representation of our system. This is a 10 by 10 world, so 100 cells, 9 types. Only 3 of these types are currently present. So the green, the teal, and the blue, no fun shapes here, just colors. The other 6 are extinct. And one more point here is that the transparency is how dense they are in the cell. So we can have different concentrations of these things in cells. And if you see a couple of those cells have a really light, just tinge of a color, that just means that the type exists in that cell, but not at a very high level. And so what actually drives the system? What do we give the system in an attempt to make it do something interesting? We break this into 2 main components, the interactions and the environment. And the interactions are just the ecological properties of these types. So how the types interact with other types. We represent this as a community interaction matrix, or as a directed graph, as you can see there. And every type somehow interacts with another type, with all of the types, either in a beneficial, detrimental, or neutral way. And that's encoded within this matrix. If that sounds super interesting and you think it's awesome and you love matrices and it really think this would drive the system a lot and you want to hear more about it, awesome. I have good news and bad news. The good news is there's an entire talk dedicated to this. The bad news is I'm not giving it. It is tomorrow morning, so given by, I believe, Emily Dolson now. So if you would like to hear more about matrices in a system like this, definitely attend that talk. But for us here, we'll be talking about the abiotic parameters, the environment that controls how these types actually interact with the world. They're up there right now. We're going to dive into them deeper in just a moment. But at a high level, they are the diffusion, the seeding, and the clearing. And we'll talk more about what those mean in just a second. So we'll start with the diffusion. Diffusion is very simply how easily these types can move about the world. So in a world with very high diffusion, easy movement very quickly can spread in one time step. And in a world with low diffusion, it's much slower. You need more time to propagate across the world. Seeding is our second abiotic parameter, and this just controls how types actually enter the world. All of the worlds will start empty and will be seeded over time. And so in a world with high seeding, we will have maybe in a single time step, a lot of seeding events in which a small amount of a type will enter the world. And in a world with low seeding, a small amount will enter a few cells. And this is analogous to like migration from a global species pool. The final abiotic parameter is clearing. We allow for these things called clearing events that will essentially just wipe a cell clean, completely empty it out. So in a world with high seeding, these events are common. Maybe in a single time step, you have a lot of cells that are wiped out. And in a world with low clearing, that is far more rare. The cell will be cleared quite a bit less. And so just a quick summary of our system and how it works. We have the three abiotic parameters, the diffusion, seeding, clear. We have an interaction matrix that controls how the types interact with each other. And these two things together will run over time and give us a final world state that will typically stabilize at some configuration. And that's what we'll call the artificial ecology world state. Of course, we're focusing on the abiotic parameters here. So how do we know which results are interesting? I told you we have these abiotic parameters in this matrix and they make this world, but how do we know that they actually connect to the major transitions that I talked about at the beginning of this talk? And to kind of examine this, we introduced the idea of transitionability. And this essentially says, how ready do we think that this world and these types are to approach a major transition? And this will hopefully make more sense when we go into how we actually measure it. But for now, we're going to simulate the two extremes and do some comparisons to hopefully make this more clear. We're going to simulate a random scenario and a built-in community level reproduction scenario to quantify transitionability. And we call them our null models. We have our random null model and our adaptive null model. And in both of these models, they act a lot like the artificial ecology that I introduced earlier. They're a grid of cells. They have types in them. The big difference is that there's no movement. So you cannot move between cells. Once you are seated in, you are stuck there. And the adaptive null model has an extra constraint that says there is a chance for probabilistic group level reproduction to occur here. I'll go more into that in a moment. For now, just the random null model, once again, no movement, each cell is completely isolated, and the contents of a cell will depend only on the random seeding events and the growth that occurs from those random seeding events. And in the adaptive null model, each cell essentially functions as a Darwinian individual. Once these seeding events can act as mutations where you get a new thing in from the outside, and selection can be imposed on the total biomass of the cell. That's what we use in this system. And so the more biomass you have, the higher probability you reproduce as a whole into another cell. And so you can see on the right here, if the circle, the smiley face and the heart do very well together, they grow to a very high biomass. they can, as a group, reproduce into another cell in the world. So we have our two null models, and we look for scenarios in which they are different. If they're the same, it's not very interesting. You can easily imagine a scenario where we have 9 types. Each type is mutualistic. The null model and the adaptive model will look exactly the same because even with no adaptation, they're going to work together, even if the cells don't move. It's just not interesting. And so we want to look for a scenario where these two models are different. The adaptive is different from the random. And we also look for a scenario where the artificial ecology looks more like the adaptive null model than the random null model. So in this example here, in the adaptive null model, our smiley faces and hearts, once again, have dominated the world. They are working together very well. But in the random null model, without movement and without that group level reproduction built in, the stars are really dominating. Maybe it's a selfish species. It doesn't play well with the others. And so it's allowed to hang around when we have our random null model. And so we look for scenarios where the artificial ecology looks a lot more like the adaptive null model. And if you want to talk more about how we turn these into numbers, feel free to come by me at some point. There's a quick recap of what we're actually doing. We want to find the abiotic parameters in the interaction matrix that facilitate types to act more like a Darwinian individual than like separate entities that are assembling at random. So to actually run our experiments here, we needed to kind of constrain our search space a little bit, trying to find interesting regions while modifying both the matrix and the abiotic parameters is very difficult. It's a problem in and of itself. And so we cut down and we used just these 4 matrices here for this work. These were identified by a genetic algorithm in a previous work that we did. We call them matrix classes, but really they're just common structures that are known to produce transitionable results. And here they are right here. We can again talk more about these at a later point if you're interested at all. So I have heat maps here. There's a lot, very big heat maps, lots of heat maps. You don't need to read all of them. You don't need to know exactly what's going on in them. I'll summarize all the points on the right. But if you do like heat maps, the darker colors represent the highly transitionable worlds, and the lighter colors represent worlds in which maybe the artificial ecology world looked like both of the models or looked like the random null model more than the adaptive null model. And so what we see is that a low clearing probability is actually consistently important. We thought that the low clearing was very important, or a high clearing probability, I'm sorry, was very important prior to this work. But it appears for these matrix structures that a low clearing was very important for most of them. Certain matrix structures were more robust to changes in abiotic parameters, and high seeding probability was actually less important than previously believed. We had an intuition that high seeding was going to be very important. That's also what the GA maximized for, was high seeding. But here that didn't seem to be the case. And we'll have 2 more slides. These are the final 2 classes. I only showed the first 2 classes in the previous slide. But essentially the same thing here again. Again, low clearing seemed to be very important, and the high seeding that we thought was necessary didn't necessarily emerge. And again, as you can see, definitely differences in how robust each of these matrices was to changes in the environment. So like class 4 there really needed a low clearing probability to get off the ground. It was struggling to make a difference. If it was anything, any clearing probability essentially in that world was detrimental. And as for future directions, we'd really love to explore evolvable interaction matrices, as well as allow for species to evolve behaviors that are detrimental to the self, but are beneficial to the community as a whole, and examine how that works. As well as more diverse matrix structures with hopefully identifiable motifs that commonly appear in worlds found to be adaptable. Thank you, and I'll take any questions.